Welcome back to the class, children. In today's class, we are going to discuss few terminologies related to plant hybridization and genetics. We are looking into the chapter Classical Genetics, and so far in the previous classes, we have discussed about the meaning for the term genetics, the subdisciplines of genetics, history of Mendel, all those things we have discussed in the previous class. Before entering into the experimental work of Mendel, let us discuss a few terminologies that are related to hybridization technique and genetics, so that it will be very much convenient to understand the process. The first terminology is self-fertilization. Self-fertilization means the fusion of male and female gametes of the same plant, or the fusion of the pollen grains, because pollen grains are the male part of the flower whereas egg is the female part of the flower. The pollen grains contains the male gametes, whereas the egg is nothing but the female gamete that is present inside the gynoecium or the ovary, right? The fusion of these the male and female gametes of the same plant or the same flower is called as self-fertilization, which is also called as autogamy. And self-fertilization, regarding the plants, self-fertilization is certain. It will be happening most conveniently, or it is sure in the plants which are bisexual. If you look at this image, so this is a bisexual flower. A bisexual flower is the one that has both male part as well as the female part. So this flower has the male part, the stamens, and the female part, the pistil. Both the pistil as well as the stamens are present in the same flower. So this is a bisexual flower. So the pollen grain which are present inside the anther of this flower, after maturation, get dispersed and it will fall on the stigma of the pistil which is present in the same flower itself. So after the pollination, then it undergoes the germination of the pollen tube and the fertilization takes place. Now the eggs or the ovules present inside this gynoecium is fertilized by the pollen grains which are present or which came from the stamen of the same flower. So this is what we call it as self-fertilization. The fusion of the pollen and egg of the same plant is called as self-fertilization, which is also called as autogamy. And self-fertilization will always lead to the maintenance of homozygosity. Homozygous condition refers to the maintenance of similarity between the alleles, right? So if the plant is self-fertilized for many number of generations, then the plant is said to be a pure breeding variety or true breeding line or pure line breed. So what is a pure line breed? A pure breeding variety is a plant that undergoes self-pollination for many generations, and it shows a stable inheritance of traits from parents to offsprings, and moreover it maintains the homozygosity. Because those plants that are undergoing self-fertilization and there is no chance of cross-pollination, and hence there won't be any recombination of genes. So whatever the character that is maintained in the plant, it will remain in the plant itself and it will be transmitted to the generations as such. So how many generations it comes, the same fertilization occurs and the same characters will get transferred from one generation to another generation. And this is what we call it as pure line breed or true breeding line or pure breeding variety. The next terminology is emasculation. Emasculation means a removal of male part of an organism. In plants, it is nothing but the process of removing the anthers, because anthers are the one that contains pollen grains and pollen grains contains the male gametes. So the process of removing the anthers from the flower of a plant is what we call it as emasculation, removing the anthers from a flower. Why this process has been carried out? So this process has been performed to prevent the self-fertilization. because while speaking about the self-fertilization, I have already told you self-fertilization is certain in bisexual flowers. So to avoid this self-fertilization, the anthers are removed manually. They are removed even before the flowers are blooming. So this process of removing the anthers is called as emasculation. After the emasculation process, there are chances for cross-pollination if the flower is kept opened. So, to avoid the undesirable pollens to fall upon the stigma and make it fertile, to avoid those unwanted fertilization, the emasculated flowers are covered with butter paper or polythene bags. So if you look at this image, the emasculated flowers are covered with butter papers to prevent the unwanted pollen grains to fall upon them, right? So this is what we call it as bagging, and usually after bagging, tagging will be taken place. So just bagging is enough now. So bagging is the process of covering the emasculated flower with a butter paper or polythene bags to prevent the cross pollination with undesirable pollen grains. The next is genes. So you know, genes are the portions of DNA. Any portion of DNA that carries information for a particular trait acts as a gene. So genes are the portions of DNA which acts as the physical and functional units of heredity. Genes are the physical and functional units of heredity. Why? Because those are the portion that contains information for a particular phenotype. So that is why they are called as physical unit as well as the functional units of heredity. And the place where the genes are located on the chromosome is what we call it as locus. If you look at this image, so these are the chromosomes, and here one gene is there. Here gene is there. So this gene G, it is represented by G, and this gene is represented by R. These are the genes which are located or distributed all throughout the chromosomes. And the place, so this is the place where this gene is located. So this is called as locus. And this is also called as locus. So we have 4 loci, right? So the place where the genes are located on the chromosome is what we call it as loci. Next is alleles. Alleles refers to the alternate forms of a gene. For easy understanding, so see this is both simply called as T, but this is called as capital T and this is called as small t, that is uppercase and lowercase. But simply we call it as T, but the T occurs in different forms. It plays a dual role, right? If you watch Onion movie, in that Onion movie, Vikram plays the role of 2 different personalities. So like that, a single gene has various forms, right? So here it is mentioned only 2 forms, 2 alternate forms. So capital T and small t. Even there may be few other forms, T1 or T2, small t1 or small t2 or T2, capital T2 or capital T3. Just for example I am saying. So the alternate forms or the different forms of a same gene, but all can be called as a T. So that is what we call it as alleles. The various forms or the versions, the different versions of a single gene is what we call it as alleles. Then homozygous condition and heterozygous condition. Homozygous, homo means similar, zygosity, zygous means the degree of similarity between the alleles. How far the alleles are similar to each other, that is what we call it as homozygous condition. So look at this chromosome. So here this chromosome has capital A allele and this chromosome is also called as capital A allele. Both are uppercase alleles. Both contains capital A. The alleles are capital A. So this condition is what we call it as homozygous. Homozygous condition refers to the chromosomal condition having similar alleles of a gene. Both are A and moreover both are capital A. That is what we call it as homozygous condition. In contrast, look at this condition. Both are A, but one is capital A, other one is small a. So this condition is called as heterozygous. It refers to the condition of chromosome having dissimilar alleles. So this is one allele, capital A, and this is another allele which is denoted as small a. Both are not same, but both belongs to same gene. It is simply called as A. But this is capital A, allelic form. And speaking about the allelic form, this is capital A and this is small a. Both are different. Different alleles present on chromosomes. So this condition is called as heterozygous. So homozygous means the chromosomes having similar alleles of a gene, whereas heterozygous means the chromosomes containing dissimilar alleles of a gene. Next, dominant allele, recessive allele. So dominant allele. So allele which suppresses or it masks or overrides the effect of another allele. So there may be 2 or 3 forms of a same gene, right? As I have already stated. So this is capital A and this is small a. Both are A, but this is capital A, one form of allele. This is small a, another form of allele. There may be another form A1. There may be even another form A3. So all are same gene but different forms, right? So now dominant allele is the allele that masks or overrides the effect of another allele of the same gene. So imagine a situation, right? For a function, there comes the CM along with his cabinet ministers. Who will be given the importance? The chief minister will given all sort of importance. Even though the ministers are there, the chief minister will be given all sort of respect and all the importance will be given, prior importance will be given only for the chief minister. Here, the chief minister dominates the effect of other ministers. Like that, the dominant allele overrides or masks the effect of another allele of the same gene. Whatever the condition may be, whether it is homozygous or heterozygous, it exhibits its phenotype, right? So that type of allele is called as dominant allele. Whereas, what is recessive allele? Recessive allele is the allele that cannot express its phenotype in the presence of dominant allele. Let us come back to the situation. So if there is CM, chief minister, and there is a cabinet of minister, a minister cannot by his own do all sort of activities without the permission of chief minister, right? Even though he has certain qualities and he has the ability to do certain activities, without the permission of chief minister, he can't do it by his own. So his activities are controlled by the effect of chief minister. Like that, so the recessive allele cannot express its phenotype in the presence of dominant allele. If there is no dominant allele, if only the ministers are attending the function, then all the priority now goes to the ministers. But in the absence of chief minister. Like that, if the dominant allele is absent or if it is not present, then at that condition only the recessive allele expresses its phenotype. So always a recessive allele will express its character or its phenotype only in the homozygous condition, right? Whereas dominant allele expresses its phenotype both in homozygous as well as in heterozygous condition. And usually dominant allele is denoted by the uppercase alphabets and recessive allele is denoted by the lowercase alphabets. So look at this condition. So this is capital A allele and this is small a allele, lowercase and uppercase. Both are A, same gene. Let us imagine these alleles are responsible for the hair color. Capital A is black, that is dominant, and small a is responsible for blonde. Blonde hair means paler. Let us imagine paler or white hair color, right? So small a is responsible for blonde hair color, right? So look at this condition. Now these chromosomes have 2 alleles and both of them are capital A. This is a homozygous condition. Now what will be the phenotype for this one means the phenotype for this condition will be the black color because both are dominant alleles, then obvious the phenotype will be a black color. Now in this condition, we have a heterozygous condition. We have a dominant allele as well as the recessive allele, but recessive allele cannot express its phenotype. Why? Because of the presence of dominant allele, right? So now in this condition also, heterozygous condition also, the color will be a black one. And the same condition, this is also a heterozygous condition because of the presence of dominant allele. Even only one dominant allele is present. Now the color also will become a black. So in all these three conditions, so here homozygous dominant and these two are heterozygous dominant. In all these three conditions, the color will be black one. Whereas look at this last image, and this is a condition where there is no dominant allele. Both the alleles are recessive. So in the absence of the dominant allele, the recessive allele expresses its phenotype and the color now will be the blonde hair color. So recessive allele expresses its phenotype only under the homozygous condition, whereas dominant allele expresses its phenotype in both homozygous as well as in the heterozygous condition. Next is genotype and phenotype. Genotype, the genetic constitution. What is the composition of genes which are present in the organism? That is what we call it as genotype. So the genetic makeup of an organism is called as genotype. Whereas phenotype refers to whatever the observable characters that are present in an organism, right? So this is a illustration. So in female we have both X chromosomes, and the X chromosome contains the genes that are responsible for the femaleness. So this is the gene makeup, and this is what the baby child, right, or the girl child is the phenotype. Because of this genetic makeup, the phenotype is female. Likewise, in male, there is one X chromosome and there is one Y chromosome. Because of this genetic XY makeup, the child is going to become a male, right? So, and the genetic constitution is what we call it as genotype, and whatever the characters what we see, what we directly observe is what we call it as phenotype. And phenotypes are determined by the genotype. That one should know, right? So phenotypes are determined only by the genotypes. So whatever the genotype is present, then the respective phenotype will be expressed. So here we have both X chromosomes. As a result, the phenotype is going to become a girl child. Whereas here one X and one Y chromosome, so the phenotype is going to be a male. So this is what we call it as genotype and phenotype. Yes, children. So today we have discussed few terminologies related to the plant hybridization as well as genetics. So here are the possible questions. What is emasculation? What are alleles? What is a pure line breed? This can be asked as this question can be asked as a 3-mark question. Define pure line breed. Right? So a pure line breed is a plant variety that undergoes self-pollination for many number of generations and maintains a homozygosity condition. So that is what we call it as pure line breed. So all these can be asked as either 2-mark or 3-mark questions. Hope this session will be a useful one. Let us meet in the next episode. Thank you.